Okay, so today we are diving deep, and I mean really deep, thousands of meters below the ocean's surface. We're on the trail of a scientific detective story, but our main character isn't a person. It's a tiny organism from the bottom of the sea that's hiding a massive secret.
So just think about this for a second. What if one single microscopic life form held the answers to some of our most terrifying medical challenges? Things like drug-resistant superbugs and aggressive cancers. It sounds like science fiction, right? Well, that exact question is what kicked off this whole incredible investigation.
So our story starts right here, in the sediments of the Mid-Atlantic Ridge. It's a crazy extreme environment, and scientists exploring down there managed to isolate this unique little red yeast. It's called Rhodotorula mucilaginosa. They had a hunch that to survive in a place like that, it just had to be cooking up some really interesting chemistry. And boy, were they right. When they got it back to the lab, they found it was making some seriously powerful stuff. But there was a weird catch. Its abilities were, well, they were all over the place. It was almost like it had a split personality.
And this is where the mystery really starts to get good. It turns out this one yeast acts like two totally different creatures depending on what you feed it. And I'm not talking about a small change here. It's a complete, radical switch in what it decides to create.
Okay, check this out. When they grow the yeast on a high-carbon diet, lots of sugar, it produces chemicals that are amazing at killing human lung and breast cancer cells. But if you switch its food to a high-salt diet, it completely changes its game and starts pumping out compounds that fight off dangerous superbugs like MRSA. So you've got the same yeast making two totally different medicines. And that is the billion-dollar question, isn't it? How on earth can one tiny microbe have two completely different chemical arsenals? It's like having a factory that can be programmed to make either an anti-cancer drug or an antibiotic. The scientific detectives had their case, and they absolutely had to figure out how this little guy was pulling it off. So, the team decided to tackle one mystery at a time. They focused in on the anti-cancer ability first. The mission was clear. Hunt down the exact molecule, the active ingredient that was actually responsible for killing the cancer cells. This meant they had to sift through every single chemical the yeast was making. And to do this, they used this absolutely brilliant technique. They basically created a social network for molecules. You see all those dots? Each one is a different chemical, and they're linked to others that have a similar structure. By laying their test data over this map, they saw one big cluster of molecules just light up. It was the prime suspect for the anti-cancer activity. And all of these prime suspects belong to one chemical family, PEFAs, which stands for polyol esters of fatty acids. Now, the easiest way to picture these is as sugary fats. So, the hunt was officially on. They had to isolate these specific PEFAs and prove, once and for all, that they were the cancer killers.
At this point, the researchers were feeling pretty good. I mean, they were confident they had their culprits. All they had to do was isolate them, test them, and, publish the paper. But science is rarely that simple, and this investigation was about to take a turn that nobody saw coming.
So, back in the lab, the team hit a new wall. Trying to pull one pure molecule out of a complex soup of really similar chemicals, it's just incredibly difficult stuff. But after a ton of painstaking work, they did it. They finally had the pure PEFA molecules in their hands, ready for the final showdown.
And here they are. I mean, just look at those names. You don't have to know what they mean to appreciate the insane level of chemical complexity they were dealing with. These four distinct molecules were the lead suspects, all cleaned up and ready to be tested one-on-one against cancer cells.
And here it is, the big twist. When they tested them, absolutely nothing happened. Zero. These molecules, the ones they were so sure were the assassins, were completely harmless to cancer cells on their own. The prime suspects were innocent. The trail went ice cold, and the mystery was suddenly deeper than ever before.
So with all their chemical clues leading to a dead end, the team had to completely switch up their strategy. They figured, okay, if the answer isn't in the molecules themselves, maybe it's in the instructions for making them. It was time to step away from the chemistry bench and dive into the yeast's genetics.
The scientists sequenced the yeast's entire genome. That's all of its DNA. This gave them the complete cookbook, every single chemical recipe the organism had. And we're talking about sifting through over 20 million base pairs of genetic code to find the answer. It's a massive amount of data.
And buried in all that code, they found it, a cluster of genes they labeled BGC3. This is what's known as a biosynthetic gene cluster. And you can think of it as a dedicated recipe book in the DNA for building one specific type of molecule.
They confirmed that BGC3 had the instructions for making the fatty acid part of the PEFAs. So they had the recipe, but it still didn't explain why the final cake didn't work.
And that's when it finally clicked. The team put all the pieces together. The chemistry that failed, the genetic blueprint they'd found, that initial powerful effect, and they came to a new and actually much more beautiful solution to the whole mystery.
Now, this quote from their research, even though it's about the bug-fighting activity, actually holds the key to everything. The scientists realized the power probably doesn't come from any one molecule, it comes from synergy, from a whole bunch of different molecules working together as a team.
So what does that mean? It means this yeast isn't making a single silver bullet. It's brewing a complex, powerful chemical cocktail. Each individual ingredient might be weak on its own, but when they're all mixed together in the precise recipe the yeast creates, they become this incredibly potent weapon against cancer.
So what are the big takeaways from this whole deep-sea detective story? Well, first, that a single organism can be a tiny, programmable pharmacy, changing its output based on its diet. Second, and this could be a huge deal for discovering new drugs, nature's real power often comes from synergy, not a single magic bullet. This kind of challenges the whole way we've been looking for medicines. And finally, it just proves that the deep ocean is this enormous, unexplored library, full of these complex chemical secrets just waiting to be found.
Which really leaves us with one last, kind of mind-blowing thought. This was just one tiny yeast from one spot in the mid-Atlantic. It makes you wonder, doesn't it? How many more cures, how many more incredible scientific mysteries are just waiting for us down there in the dark, unexplored corners of our own planet?